All right, so this is the first part of chapter 13, which has to do with cardiac physiology. We're going to talk about the functions and components of the cardiovascular system and lymphatic system. A lot of this is going to be review from anatomy. I will review some of them. I may not go into uber detail in a lot of it because it should be review, but there is a lot of information in your slides, so I'll leave that there for your so you can read it. The major part here is definitely transportation of oxygen or other respiratory gases, nutrients like glucose or fats, hormones, and waste products. There is regulation occurring here. We know that our hormones travel in our blood. In anatomy, or we're going to discuss today, temperature, blood can regulate temperature as well. So there's regulation occurring here. And you should be aware from anatomy that there is a protective mechanism with our immune system function from our white blood cells and our lymphatic systems, as well as blood clotting. Blood clotting is a protective mechanism. So, we're going to largely focus on the cardiovascular and lymphatic systems. First, the first 80% of this lecture is going to be on the cardiovascular system. And it comprises of blood, the heart, which is our pump, and then the blood vessels, which are literally just the tubes that carry our blood from system to the heart. Then we will talk about the lymphatic system briefly, and I'm not going to get into it right now because we'll get into it when we discuss it. So our blood is considered an organ. If you can remember from anatomy, it is a connective tissue. It's a liquid connective tissue. And it is what allows for transportation of our respiratory gases, movement and delivery of nutrients, hormones, and waste, temperature regulation, clotting, immune system function via our white blood cells. When you centrifuge blood, the heavier bits get to the bottom, and those are our red blood cells. The lighter, the less dense stuff goes to the top. That is our plasma. And then in the middle, there is a thin white layer called the buffy coat. The buffy coat is made up of white blood cells and platelets. So what you can see is that, white blood cells only make up like 1%, less than 1% of our blood composition. If you were to take an average person, their total blood volume is about 5 liters. And so, looking at this, it's about 45% formed elements, 55% blood plasma, and about 5 liters total in an individual. So the plasma is this fluid right here, which consists of ions like sodium, potassium, calcium, hormones, maybe things that we metabolize like glucose, proteins, nucleic acids, antibodies, and even waste products like drugs. Maybe someone can take a drug test from giving their blood. Red blood cells make up the majority here, and as a whole, when you centrifuge it and you get this out, this is called the hematocrit. So hematocrit is about 36 to 46% in females, about 41 to 53% in males. There are a few different types of plasma proteins in our blood. The 3 types that we will talk about are called albumins, globulins, and fibrinogen. Albumin is the majority of it. It accounts for about 60 to 80% of the plasma proteins, and it maintains it stays within our plasma. And so say I was going to draw our plasma here. And it creates this colloid osmotic pressure. Say that, maybe this is some different fluid here, interstitial fluid of the body, where the albumin is going to draw water towards it from our capillaries or from other areas. So I showed the example of interstitial fluid here. This is going to draw water towards it, and so this helps us maintain our blood volume as well as blood pressure. It helps maintain this osmotic pressure, and it's called colloid because that has to do with proteins. So that's the job of albumins, and we have a lot of albumin protein in our blood. Globulins, their job is to carry lipids. We should know that lipids are hydrophobic, and they don't dissolve well in water, right? They dislike water. And so for them to travel in blood, they need to attach themselves to a protein, and they do via these globulins. Fibrinogen is a protein that serves as a clotting factor. It is going to convert into fibrin, which we're going to talk in a little bit. It impacts clotting, but I'll talk about the physiological components of these slides. First off, we're going to talk about how red blood cells are produced. But one big thing is that they do not contain a nucleus and a mitochondria. They don't contain any organelles at all. And so they do not do mitosis. They cannot do mitosis. They are produced in our red bone marrow, which will be in a future slide. And they only last about 120 days. And so that means they don't last forever. And where they are destroyed is in the liver, the spleen, the bone marrow. And maybe you've seen individuals who have liver damage or cirrhosis of the liver. And these individuals might have jaundice. If you've seen that before, it's really striking. You get yellowing of the skin. That yellowing of the skin and the whites of the eyes, that's like a really big telltale sign, is caused by a protein called bilirubin. Bilirubin is destroyed by our liver, and it is the leftover protein from red blood cells, and it is pigmented. It looks yellowish, which is why if you have a buildup of bilirubin, it expresses itself on the skin. So leukocytes, in comparison to the erythrocytes, they are full cells. They have nucleus, they have organelles, and they even have this amoeboid ability. And what that means is that they can move around like an amoeboid. They can grasp things like an amoeboid, and they have this phagocytotic capability too. They can actually do something really interesting where they can squeeze in and out of capillary walls. This is called diapedesis, and that allows them to go to the specific area of location, specific area of infection. They can move around to specific areas of infection because they are the cleanup crew. We can separate these in granulocytes, and these top three ones are granulocytes. They have these granules. They may initiate inflammation. They can help detoxify. They can help phagocytose. And then these two down here are called agranulocytes. They do not have granules in them like the upper two. Agranulocytes this is not perfect. So lymphocytes produce antibodies. where monocytes are phagocytic. So both of them don't do both. Actually, there's a couple lymphocytes that can phagocytose, but monocytes do not produce antibodies. Only lymphocytes produce antibodies. Lymphocytes are going to produce B cells and T cells. Maybe you've heard of those before. And B cells are what produce our antibodies. Platelets are actually cell fragments. These are actually cell fragments. They are the smallest of the formed elements. And they can also move in an amoeba-like manner. And they are going to assist with blood clots. They also release serotonin, which can cause vasoconstriction of an area, and that helps reduce the blood flow to assist in blood clotting. They secrete growth factors to help maintain the integrity of the blood vessel wall, and they only survive for about 5 to 10 days. Hematopoiesis is the official name of blood cell production from stem cells. This occurs in our bone marrow, specifically in our red bone marrow, as well as lymphoid tissue. So our red bone marrow produces about 5 billion blood cells per day. In the fetus, not a baby, but in the fetus, it also occurs in the liver. I will share that in children, in young babies, they have red bone marrow everywhere in their bones, in the in the medullary cavities, also called the marrow cavities of their bones. Adults, however, do not have red bone marrow everywhere. It's really only found in our spongy bone. And so this is limited to, the epiphyses of our long bones or in our flat bones. And so we don't have as much red bone marrow tissue as children do. So there's 2 types here. Erythropoiesis is the formation of erythrocytes, which is the, red blood cells. And it's stimulated by a hormone called erythropoietin. Erythropoietin is released from the kidney, and it's going to stimulate production of red blood cells in our red bone marrow. Leukopoiesis is the formation of leukocytes, which would be our white blood cells. And it's stimulated by a few different things, which we call cytokines. Cytokines are just like chemical messengers, kind of like hormones are, but they act in a different way. So these are going to be autocrine regulators secreted by the immune system. If you can remember what autocrine meant, it means they release, and they act on the same cell that releases it. Thrombopoietin is an example of a hormone that stimulates the growth of megakaryocytes, a type of cell, which are going to develop and produce platelets. So erythropoiesis, we have a lifespan of 120 days for our red blood cells, and about 2.5 million red blood cells are produced per second. Old red blood cells are removed by phagocytotic cells in the liver, the spleen, and bone marrow. And iron is recycled from these red bone marrows from this area back into hemoglobin production back in our red bone marrow. I wanted to point out Lance Armstrong, or even other athletes who get caught for cheating. If you don't know who Lance Armstrong is, he was he won 7 Tour de France's, but those actually got revoked when he admitted to cheating. And what he took was erythropoietin. He did blood transfusions, which means he injected more red blood cells into his body. And then there were a few other hormones that he just did. But, you know, this erythropoietin was added so that he could include or really ramp up the number of red blood cells in his body. Athletic institutions check for doping, and they check by taking a vial of their blood, centrifuging it, and making sure that the red blood cells make up about 45% of the composition, right? And so what did he do? He also infused blood plasma to kind of try and level out that concentration. So some clinical examples of this, you've probably heard of anemia. Individuals who are iron deficient have a type of anemia where they have a low red blood cell count because their hemoglobin concentration is low. This is a very common form of anemia. It impacts females way more than it impacts males. And so it's really important to eat iron-rich foods like greens, like spinach, or if you need to, take an iron supplement. This is why blood kind of has that brassy, kind of metallic smell. It's because of this iron in our hemoglobin. Hemoglobin needs iron to be able to bind to oxygen. And so you need to make sure that you have a diet rich in iron. Pernicious anemia is the lack of intrinsic factor, which is produced by specific cells in the stomach. And that's needed for vitamin B12 absorption. And then aplastic anemia is due to bone marrow damage because of chemotherapy and radiation. And so this is actually kind of common with cancer patients. Polycythemia is when you have an abnormally high red blood cell count, and this could be due to being in high altitudes for long periods of time where there is low oxygen content. And so what your body does is it takes about 10 days for the body to acclimate to these high altitude. And it's because we release erythropoietin and we increase our red blood cell count to be able to pick up more oxygen molecules that are lacking in that high altitude environment. Leukopenia is an abnormally low white blood cell count, often due to that radiation and chemotherapy that was similar. Leukocytosis can be an abnormally high white blood cell count, and this can be due to infection. When we have infection, our body goes ham and our white blood cell count increases significantly to try and stave off that infection. Leukemia is cancer of bone marrow, and you may have a high number of white blood cells, but these white blood cells may be abnormal. Blood clotting is defined as hemostasis, which is the cessation of bleeding or the stopping of bleeding when a blood vessel is damaged. When we damage a blood vessel, that's what causes our blood to be released from the blood vessel, which is what causes bleeding. Blood clotting is going to be the stopping of that. And how this works is that when you damage a blood vessel, like damaging the endothelium of our vessel wall here, it's going to expose collagen fibers. And these collagen fibers are exposed to the blood. This is going to cause vasoconstriction. The blood vessels are going to literally constrict and reduce the blood flow to the area. It's going to sends signals to platelets, and the platelets are going to collect at that open damaged area, and that is going to cause production of fibrin. So the platelets bind to collagen. The platelets recruit more platelets, forming a platelet plug. And by the way, this is positive feedback, an example of positive feedback. And it does this by secreting serotonin to vasoconstrict the blood vessel, ADP, which makes them kind of sticky. It kind of acts as like a chemical signal to bring platelets near them. And then thromboxane A, which kind of does the same as ADP and serotonin. So they glue together in this positive feedback manner and plug up the damaged vessel, and this continues until it is completely plugged. Fibrinogen is one of those proteins that we find in our blood. It is converted to fibrin, which is going to assist in this clotting mechanism, by one of two pathways. There is an intrinsic pathway, meaning when fibrinogen gets exposed to collagen, it will convert to fibrin right away, which will activate a cascade of other blood factors. Or it can be extrinsically by tissue thromboplastin in a more direct pathway. Calcium and phospholipids from the platelets are going to convert prothrombin to an active enzyme called thrombin. This is another protein that's found in our blood, and that is going to convert fibrinogen to fibrin. So prothrombin and thrombin are known as clotting factors, and vitamin K, which we can receive from our diet, is needed by our liver to produce these clotting factors. We have three major types of blood vessels here: arteries, capillaries, and veins, and they have different structures as well as different functions. You should remember that arteries always bring blood away from the heart, as in this picture, and veins always bring blood toward the heart, as in this picture. Capillaries are the site of gas exchange, and gas exchange here is completely passive, so it would be diffusion, passive diffusion. Notice the difference in structure of our arteries and veins. They have their three major layers: the tunica intima or interna, tunica media in the middle, and tunica externa. The tunica interna, we would expect our thin, simple squamous epithelium plus some connective tissue, because we always have connective tissue wherever we find epithelium. The tunica media is going to have some muscle, specifically smooth muscle, with it, along with connective tissue. And then the tunica externa is a large connective tissue layer. The most variable layer between our arteries and veins is the tunica media. Notice for the arteries, the tunica media has way more muscle in the artery than compared to our vein over here. And notice for capillaries, they only have one of those layers. In the capillary, we only have the tunica interna, and that's it. Only that simple squamous layer with a little bit of connective tissue. Blood pressure is at its absolute highest when blood is coming off of the heart. And that's why arteries are more muscular and they're more elastic. They have to withstand that high blood pressure, and also because they hold a really important job to send oxygenated blood to the different parts of the body. There is a lot of muscle in the tunica media and elastic fibers in the connective tissue layers, in the 3 connective tissue layers. So there's a lot of elastin here, a lot of elastic fibers. This allows for expansion during systole, but also recoil during diastole. And it helps maintain for a smooth blood flow. So the arteries that are closer to the heart are more elastic because they have to withstand that higher blood pressure. As you get further away from the heart, but you're still an artery, they actually gain in muscle because they have to be able to pump blood to those important tissues for oxygenation to occur. They have more muscle here, they have less elasticity, so they are less distensible. Arterioles actually cause the greatest pressure drop, and most of these are going to connect to our capillary beds. Some connect directly to veins to form an arteriovenous anastomosis. So our capillaries are our thinnest and narrowest, but the most numerous of blood vessels. They only contain the tunica interna with their single layer of endothelial cells. This allows for a lot of surface area for gas exchange to occur, and it's in these capillary beds that gas exchange occurs. And we're not just talking about O2 into the tissues, but we're also talking about carbon dioxide waste being removed from tissues as well. Blood flow through a capillary can actually be changed. It can actually be shunted, and that is by these opening and closings of these precapillary sphincters, which are kind of like these doors that can either open or close and say whether there will be blood flow through a capillary bed or not. And let me give you an example. Say I'm actually going to Tahoe this weekend, and say it's really, really cold outside and I don't have mittens on. Maybe you've experienced this before where your fingertips go numb, or your tips of your toes go numb when it's really cold outside. And that can be because of a temperature regulation mechanism where your precapillary sphincters are going to close off the blood supply to your extremities, like your fingertips, so that heat will not be lost in those areas, because it's in the areas of your extremities that you can lose heat the most. And so your capillaries decide to shunt blood from those tissues for a little while. And that's why you get numbing of the fingers when you're in cold environments, and maybe they look pale. It's because there's no blood flow, or that blood flow is getting shunted for a little bit. There are different types of capillaries. In continuous capillaries, they're called continuous because the endothelial cells of the simple squamous epithelium are continuously joined together. They are tightly joined, and they're going to have some narrow channels that can permit exchange between them to allow for gas exchange. So we see these in our lungs, we see these in our muscle, we see these around in our adipose tissue. In fenestrated capillaries, they actually have these bigger pores, and so they're very permeable. And so you'd expect this where we have a lot of maybe secretion of molecules going on, or maybe filtration of molecules going on. And an example of that would be in your kidneys. There's a lot of filtration and secretion going on in your kidneys. In your endocrine glands, remember that's where hormones are secreted into the blood, a lot of secretion going on. And in the intestines, where we have a lot of absorption of nutrients going on, or movement of nutrients. Lastly, we have discontinuous capillaries. Discontinuous capillaries have the biggest of gaps in them, and they are big, they are leaky, they are discontinuous in that way because there's big gaps in between them. And notice where we find them. We find them in the liver, in the spleen, in the bone marrow. Wherever we're going to have blood cells moving in and out. We have bone marrow producing blood cells, so that's where, red blood cells or white blood cells can be moving in and out. And in the liver and spleen, this is where they get destroyed, right? And so they'll be moving in and out here too. So in these areas, we have discontinuous capillaries. Veins are very different from arteries, and it's actually in our venous system that the majority of our blood is at any given time. And that's because blood pressure is really low here. Blood pressure is low in our veins. It actually does not have a high enough blood pressure to be able to push to the heart on its own. And so what it uses is the action of the contraction of skeletal muscle. Some of our veins have these valves, and these valves prevent backwards flow of blood. Remember that blood is always moving in a one-way street. And even here, it's showing blood is moving this way and only this way. We don't want backwards flow, which is why we have these valves. When we contract our muscles, it helps push blood through our venous system because the blood pressure is so low here. And so another reason why, people say exercise increases circulation. It does increase circulation in other ways, but this is a big way in which when we contract our skeletal muscles, it does cause movement of our venous blood. And our lymphatic system is kind of like a catcher of our plasma fluid. It catches plasma and it transports this interstitial fluid back to our blood after it has cleansed it in a way. So it also, it has this transport mechanism. It has this cleanup crew mechanism by providing this immunological defense. And one other thing is that absorbed fats that we eat are too big to travel in the blood. Like in our villi, we're going to have fat absorption here. These fat globules are too big to go into the capillaries in the villi of the small intestine. And so where they actually go are into these vessels called lacteals. And so which are lymphatic vessels. So our lymphatic system also transports digested fats from our small intestine to our blood via the lymphatic ducts. So if you can see in this picture here, there is high blood pressure in our arteries, and there is even, a higher, maybe I should write, there is high blood pressure in our arteries, and there is still high blood pressure in our capillaries. And what that's going to do is it pushes out plasma. It pushes out plasma into these intercellular tissues. And so these lymphatic capillaries are going to be able to catch the excess interstitial fluid in their capillary system, and then it'll travel throughout the lymphatic duct system. So lymph is carried via these lymphatic capillaries to lymph ducts and lymph nodes where the cleanup crew is going to be. And when I say cleanup crew, really it's just a collection of lymphocytes, like our T cells and B cells, to make sure that there are no foreign invaders, bacteria, viruses, and if there are, they will clean it up right then and there. This reminds me of, like, on New Year's Eve, how there might be a checkpoint to make sure that no drunk drivers drive past a certain area. If you don't know what I'm talking about, police will just set up at an intersection, maybe a busy intersection, and they give everybody that passes a breathalyzer. And if you pass, you can go. If you don't, you get a DUI and you go to jail. And this is kind of the same thing that occurs here. This interstitial fluid, this lymphatic fluid from the plasma of the blood, has to pass through these lymph nodes, which are collections of lymphocytes that are going to make sure that no foreign invaders are trying to pass.